Hello everyone, I am Marquisa C. Gaugay and welcome to Analytical Chemistry subject. And for the first laboratory activity in this subject, we're going to define or look the laboratory apparatuses and their uses. Let's go. But first, let's give a short definition of this analytical chemistry. So the branch of analytical chemistry deals with extracting such information from the substance by subjecting it to a certain investigative methods. For the objective in this activity, lab equipment can have dangerous side effects if not handled properly. And through this activity, we'll define the proper use of several important pieces of laboratory equipment. So let's begin. And the first material or equipment used in analytical chemistry is the graduated cylinder. A graduated cylinder is used routinely for measuring volume and is considered more accurate than a beaker because of the permanently marked incremental graduations incorporated in the clear cylinder. And the second apparatus is a volumetric flask or measuring flask or graduated flask. A volumetric flask is a piece of laboratory apparatus, a type of laboratory flask calibrated to contain a precise volume at a certain temperature. Volumetric flasks are used for precise dilutions and preparation of standard solutions. And third material used in analytical chemistry is the Erlenmeyer flask. Erlenmeyer flasks are used to contain liquids and for mixing, heating, cooling, incubation, filtration, storage, and other liquid handling processes. Their slanted sides and narrow necks allow the contents to be mixed by swirling without the risk of spills, which is useful for titration and for boiling liquids. And number 4 is the boiling flask. The boiling flask are used for heating and boiling of liquids. The flasks are designed to have round bodies with long, thin necks to facilitate uniform heat distribution and minimize evaporation. And the fifth apparatus is the volumetric pipettes. Volumetric pipettes are designed to very accurately transfer a specific amount of solution. These pipettes can only be used to deliver the volume of a liquid for which it is calibrated. Volumetric pipettes have narrow tips and a bulb-like expansion in the middle. The next one is the Mohr pipette. Mohr pipette, also known as the graduated pipette. It is a type of pipette used to measure the volume of a liquid dispensed, although not as accurately as the volumetric pipette. This uses a series of marked lines, as on a graduated cylinder, to indicate the different volumes. Now, let's proceed to the serological pipette. The serological pipette is a laboratory instrument that transfers liquids measured in volume by milliliters. The use of serological pipette is an efficient and a gentle means of mixing cell suspensions. The next one is the Eppendorf micropipettes. Eppendorf pipettes are a common laboratory tool used to measure and transport liquid. And the ninth apparatus is the Barrett or a glass head value Barrett. A Barrett, also spelled Barrett, laboratory apparatus used in quantitative chemical analysis to measure the volume of a liquid or a gas. It consists of a graduated glass tube with stopcock, turning plug, or spigot at one end. And last but not the least, or the thin laboratory material is the Barrett or a Teflon value Barrett. A Barrett is a volumetric measuring laboratory glassware which is used in analytical chemistry for accurate dispensing of variable and for measuring the volume of a liquid, especially of one of the reagent in titration. And those 10 laboratory materials is very important in analytical chemistry. And for conclusion in this activity, we should always know our lab equipment well before we do in any type of experiment, because without the proper knowledge of our equipment, we will not know how to use our materials or how to correct a mistake that we could make with our equipment. And that's the end of my video. I hope you learned something. Once again, I am Markiza Gaugay.